Hello students, in this session we will discuss chapter Biotechnology and its applications. Okay, students, every year from this chapter in NEET exam, you will find 4 or 5 questions. Here I'll highlight some key points from NCERT. You can open your NCERT book and underline these, or you can focus on these points. Okay, first page in this chapter. Here they are talking about uses of biotechnology. Uses of biotechnology. They are saying biotechnology is useful in biopharmaceuticals. Biopharmaceuticals means we can produce medicines, or we can produce therapeutic products which are used in treatment. They are saying it is useful in biological. Biological means product of biological origin. Okay, through biotechnology we can prepare many medicines, many other products. This biotechnology is useful in therapeutics, diagnostic. Diagnostic means it is used in diagnosing some diseases. It is useful in producing processed food. Processed food means nutrient-rich food. And biotechnology is also used in bioremediation. Bioremediation means use of genetically modified organism or use of organism to control pollution. One example in this is use of this bacteria, Pseudomonas putida. Pseudomonas putida bacteria, this is genetically modified and it has capability to control pollution due to oil leakage. Okay, pollution due to oil spill is controlled with the help of this Pseudomonas putida. It is a modified bacteria and it is modified through this technique. It is called recombinant DNA technology. Now you can focus on next point. Biotechnology is useful in waste treatment, energy production. Energy production means like production of long chain hydrocarbon or alcohol production. And next paragraph, they are talking about 3 critical research areas in biotechnology. 3 critical research areas. What are 3 critical research areas in biotechnology? First research area is providing best catalyst. Catalyst here may be modified organism, improved organism. So catalyst in this may be improved organism. It means it is having gene, desired gene or gene which is required to produce some particular product. Okay, that gene is present in this organism. Or this catalyst may be enzyme. Okay, or it may be pure enzyme. Now next field of research is creating optimal condition through genetic engineering for a catalyst to act. Okay, you can recall you have studied preparing bioreactor. So in a bioreactor we can grow genetically modified organism. Okay, that genetically modified organism synthesize particular enzyme and that enzyme can convert raw material into product. Okay, so second area of research is providing or creating optimal conditions through genetic engineering for a catalyst to act. And third research area is downstream processing. Downstream processing, these are techniques through which we separate and purify product. So what are how many critical research areas they mentioned in your book? 3 critical research area. In first critical research area, we focus on preparing catalyst, and in second, we focus on preparing optimal conditions for action of this catalyst, and then we focus on downstream process technology or technology which is used to purify product. Now in the next paragraph they are talking about 3 options that can be thought for increasing food production. What are 3 options given in your book? One is agrochemical based agriculture. It means we will use herbicide, insecticide, chemical fertilizer. Okay, you know there are many harmful effects of these chemicals. Now second area or second method is organic agriculture. In organic agriculture we use biofertilizer, we use biocontrol, we are not using chemicals. Now third is use of genetically modified crops. Use of genetically modified crop. This is third method of crop production. Now in next paragraph they are talking about green revolution. You know during green revolution food production was increased many times. Okay, so here they mentioned this food production was partly due to use of improved crop varieties and mainly due to use of better management practices. So there are 2 points in this, one partly and second mainly. Okay, partly due to use of improved crop varieties and mainly due to use of better management practices. Now, in the next paragraph, they're talking about tissue culture. This is important paragraph. This is important paragraph in this chapter. Okay, from tissue culture, they have given questions many times in NEET exam. You will find many questions related to tissue culture. Okay, through tissue culture, we can prepare complete plant from single cell. We can prepare complete plant from small tissue. So first here is, what is explant? Explant is any part of the plant used in tissue culture. It can be single cell or it can be tissue. It can be cell from leaf. Explant can be cell from stem. Explant can be cell from root. Part of the plant used in tissue culture is called explant. Okay. Now from this explant or this single cell, you can generate complete plant. This capability of a single cell to generate complete plant is called totipotency. Okay, this question was given in NEET exam. What is totipotency? Capacity to generate a whole plant from any cell or explant is called totipotency. Now, this cell which we have isolated from plant or explant, we will grow this cell in culture medium, nutrient medium. Now, next they are talking about components of nutrient medium. They have mentioned here carbon source in this nutrient medium is sucrose. Okay, in nutrient medium, in this culture medium, tissue culture medium, we use inorganic salt, vitamin, amino acid, and growth regulators like auxin and cytokinin. Okay, through tissue culture from single cell we can prepare many plants. We can prepare thousands of plants or millions of plants from single cell in short period of time. So this technique is called micropropagation. Okay, this question was given in NEET exam. What is micropropagation? Production of thousands of plants through tissue culture is called micropropagation. Now, plant that we have produced through tissue culture, they are genetically similar among themselves. They are genetically similar to the parent. So they are called soma clones. Now this is next question. Multiple times this question was given in NEET exam. What are soma clones? Soma clones are genetically similar plants, okay, produced through tissue culture. Now, in the next paragraph, they are talking about production of virus-free plant. Production of virus-free plant. This again from this paragraph in NEET exam, they have been questioned many times. Okay, how to produce virus-free plant, how to recover virus-free plant or disease-free plant from through tissue culture. Now they are saying if a plant is infected with disease or pathogen, plant is having pathogen. From that plant, you can isolate meristematic cell. You can isolate meristematic cell from apical meristem. You can isolate cell from axillary meristem. Then this meristematic cell can be used in tissue culture. Meristems are generally free from viruses. These can be used in tissue culture to generate new plant. Okay, so this technique is called meristem culture. Meristem culture. Question is how to produce pathogen-free or pathogen-free or disease-free plant through tissue culture by using explant from apical meristem or axillary meristem. Apical meristem, axillary meristem. Okay, these are source of explant. So what is source of explant? If you want to prepare virus-free plant from disease plant, source of explant is apical meristem or axillary meristem. Okay, we can prepare healthy plant by this technique. This technique has been used. Okay, this has been used in banana, sugarcane, and potato. Now, in next paragraph, they are talking about somatic hybridization. Somatic hybridization. Okay, this is related to tissue culture. Somatic means vegetative cell of the plant. You can fuse two vegetative cells of the two different plants. You can isolate cells from leaf or other part of the plant of two different species or two different genera, and in artificial condition you can fuse these cells. Okay, so first you have to select parent. Then from that parent cell you have to isolate that from parent plant. You have to isolate cell, and then you have to remove cell wall. Okay, if you are removing cell wall from plant cell, it is called protoplast. What is protoplast? Protoplast is a plant cell without cell wall. How we can remove cell wall? We can remove cell wall by treating plant cell with cellulase, pectinase, hemicellulase. Now remaining component is called protoplast. We can isolate protoplast from two different plants and we can fuse these protoplast. Okay, so first by digesting their cell wall, we can prepare protoplast. This was given in NEET exam. Okay, and this isolated naked protoplast, it is with membrane without cell wall from two different varieties, two different genera. If they have desirable character, then we will use protoplast. We can fuse them. Okay, we can prepare hybrid. Now product formed by fusion of somatic cell or protoplast of two different varieties of the plant is called somatic hybrid. Again, this is question for you. What is somatic hybrid? Somatic hybrid, how they are prepared? How somatic hybrids are prepared by fusing protoplast of two different varieties. So first we have to prepare protoplast, and then we can fuse protoplast and we can prepare new plant. You can see this flow chart. In this flow chart, okay, here this is potato plant and this is tomato plant. Now scientists already they have prepared this type of somatic hybrid from plants. Scientists they isolated a protoplast of tomato, they isolated protoplast of potato. These are protoplast. In artificial condition, they fused protoplast, they prepared somatic hybrid. Now this cell was used to generate new plant, and this plant is called pomato. It is called pomato. Okay, it is a somatic hybrid. So it means it is producing both tomato and potato. Okay, now you can see in this paragraph here they have used term somatic hybridization. What is meaning of somatic hybridization? Somatic hybridization is a procedure or technique of fusing protoplast of two different plants or two different varieties. You can fuse protoplast with the help of this type of chemical, polyethylene glycol. In short form, you can write PEG, or you can fuse it with the help of sodium nitrate. Okay, now product is called somatic hybrid. So here in your book they are talking about pomato. Pomato, they mentioned here pomato. How pomato is produced? Pomato is produced by fusion of protoplast of tomato and potato. They are saying unfortunately this plant did not have all desired combination of the characters for its commercial utilization. So it is not commercially successful. Now students, in next paragraph they have given here advantages of genetic modification. They are talking about genetically modified organism, and they have mentioned here genetic modification. Genetic modification has created many types of plants. Genetic modification has created crops that are more tolerant to abiotic stress. Okay, it means they prepared plants which can tolerate low temperature, which can tolerate moisture or water deficit condition. Plants, these genetically modified plants, they can tolerate high salt content, they can tolerate heat or high temperature. Okay, this type of crops are already prepared by scientists. Now second advantage they have mentioned, through genetic modification scientists, okay, they have created crops which are less dependent on chemical pesticide or herbicides or weedicide. So they have created crops, and this reduced dependence on chemical pesticide. This reduced reliance on chemical pesticide. It means dependence. Reliance means dependence. Okay, for example, pest resistant BT cotton. Okay, you know BT cotton is transgenic plant. It is genetically modified plant, and BT cotton is resistant to bollworm insect. Okay, if bollworm try to infect this cotton plant, it will not survive on cotton plant. So to control bollworm, there is no need to spray chemicals. Okay, that's why they are saying reduce reliance. But remember, this type of plant is not resistant to all types of insect pest. Now next advantage they have given, helped to reduce post-harvest loss. In this one example is flavor saver tomato. Okay, flavor saver tomato. What is flavor saver tomato? It is a transgenic plant. Okay, now you know tomato, we cannot store tomato for many months or many weeks. We cannot store this. But scientists they have created one tomato variety in which they have delayed fruit ripening, and this tomato is called flavor saver tomato. So in this they have delayed ripening. You can store this for many weeks. Okay, so this type of crops can be produced by genetic modification. Now next is increased efficiency of mineral uses by plant. This prevent early exhaustion of fertility of soil. Scientists they have created genetically modified plants which use less minerals from soil and produce high, you can say they give high yield. They give high yield, but they use less minerals from soil. So they use minerals from soil efficiently. It means they will not waste minerals. Okay, these are used in seed production, and plants it is producing seed. It is producing numerous seed, but from soil it use less minerals. So here you can focus on this. They are saying this prevent early exhaustion of fertility of soil. So how early exhaustion of fertility of soil is prevented? By reducing excess uses or excess absorption of the minerals from soil. Now next advantage they have given, scientists they have created crops which have high nutritional value. And in this one question is, one example is golden rice. Now in a NEET exam you will find question related to golden rice. Many times they have asked question on golden rice. Golden rice is transgenic plant, and it synthesizes keratin, and this keratin can synthesize vitamin A. It can synthesize vitamin A. So here they mentioned golden rice. It is vitamin A enriched rice. Okay, it can be used to overcome problems related to night blindness. Okay, or it is due to deficiency of the vitamin A. Okay, so what is example of what is nutrient present in golden rice? It is carotene, basically pro-carotene, or it is a derivative of the vitamin A. Now students, in next paragraph here they are talking about tailor-made crops. Tailor-made crops. What is meaning of tailor-made crops? Tailor-made crops means these are crops which are created or designed according to the need of the factories or industries. Okay. In many industries there is high demand for starch. There is high demand for fuel or raw material for pharmaceuticals or medicine. Now scientists, they have created crops which provide starch, or which provide fuel, or which provide pharmaceuticals. So these type of crops which are prepared according to the need of the industries, these are called tailor-made crops. Tailor-made crops means they are created according to our requirement. These are created according to our requirement through recombinant DNA technology. Now students, in next paragraph they are talking about pest resistant plants, and one example in your book is BT cotton. BT cotton is a pest resistant plant. BT cotton is generally infected by, this insect pest. It is bollworm. Okay, bollworm is a insect and it can destroy cotton plant. Now scientists, they have created cotton plant and it is resistant to bollworm. So how they created this? They discovered one gene in bacteria, Bacillus thuringiensis. Okay, BT in BT cotton refers to Bacillus thuringiensis. Okay, it refers to Bacillus thuringiensis. Okay. Now in this Bacillus thuringiensis bacteria, it contains toxin gene. Okay, it is called BT toxin gene. Scientists, they have cloned, you can focus on this line. Scientists, they have cloned this gene from bacteria and it is expressed in plant to provide resistance to insect. Which insect? Here we are talking about BT cotton, and BT cotton is resistant to bollworm insect. Okay, they have cloned this gene from bacteria and expressed in plant to provide resistance to insect. Okay, so now this plant BT cotton, this plant, okay, it is resistant to bollworm. If bollworm is feeding on cotton, then cotton is having toxin. That toxin is activated in alimentary canal of the bollworm and it kill bollworm. So you can say this BT cotton is behaving like biopesticide, not for all insect. For bollworm, it is behaving like biopesticide. So they are saying this BT cotton, okay, this BT cotton is like biopesticide. Okay, BT cotton is a transgenic or genetically modified plant. We grow cotton for cotton fiber. Okay, we are not growing this to kill insect pest. Okay, but by transferring BT gene into BT cotton, this BT cotton secretes toxin. Now this toxin will kill bollworm. If bollworm is feeding on BT cotton, bollworm is killed by this. So that's why commonly you can say it is a biopesticide. It's a biopesticide. So like this, scientists, they have created many crops. Okay, now in NEET exam many times they have asked question on this BT cotton. BT cotton is resistant to insect. Which insect? Bollworm. And now in next paragraph they are talking about insects. These are lepidopterans, dipterans, coleopterans. These are insects. These can be controlled by this BT toxin gene. Okay, BT toxin gene. But remember, in BT cotton, it is a gene for resistance, okay, to bollworm, not all insect. Okay, so they are saying all these types of insect, tobacco budworm, armyworm, or beetles, or flies, mosquitoes, all these can be controlled by this type of toxin. But in BT cotton, it is for bollworm. Okay. Now they are saying this bacteria, Bacillus thuringiensis, this bacteria produce crystal protein or toxin protein during a particular phase. What is that phase? It is called sporulation. Okay. This Bacillus thuringiensis bacteria, it produces spore. It produces spore during particular phase. What is that particular phase? That is called sporulation. It means during spore formation. So that toxin is present in spore. Okay, toxin is also called crystal protein. It is insecticidal. Insecticidal means this toxin can kill insect. Now they are saying this protein is synthesized inside bacteria. Why does this toxin not kill the bacillus? Why this toxin not kill the bacillus? They are saying inside the bacillus bacteria, this protein exists in inactive form. This inactive protoxin is not harmful. Okay, so when it is activated, this inactive protoxin is activated in alkaline pH. Where it will find alkaline pH? Alkaline pH in mid gut of insects. Okay, this is important question. Many times in NEET exam they have asked question. It is activated in what type of pH? It is alkaline pH. Now after activation, if this protein is activated, now this toxin protein bind to the surface of the mid gut. Okay, mid gut epithelial cell. Again important point. Okay, PYQ. It attached to mid gut epithelial cell. It create pore, cause swelling and lysis, and finally death of the insect. You can see question on this paragraph. Okay, it create pore. Where it create pore? In midgut epithelial cell. Where this toxin bind? A toxin bind in midgut epithelial cell. Okay, what type of pH activate this toxin? Alkaline pH. So all these are questions. Okay. Now you can see next page. Here they talk about different types of genes present in Bacillus thuringiensis. Bacillus thuringiensis may contain Cry1AC, Cry2AB genes, or it may contain Cry1AB gene. Okay, there are many other types, but in your books they have given only 3 types. They are saying toxin produced by this type of gene, Cry1AC, Cry2AB. This toxin kill bollworm. Okay, so these genes are introduced in cotton and this kill bollworm. Now they are talking about one more type of gene, Cry1AB. Cry1AB. This type of gene, it produces toxin, and that toxin kill corn borer. So it is used to prepare BT corn, and these are used to prepare BT cotton. Okay, BT cotton. What type of genes are present in BT cotton? Cry1AC, Cry2AB. What type of gene present in BT corn? Cry1AB. Okay. Previous year question. Now here you can see. So they are saying what choice of gene depends upon the crop and the targeted pest. Okay, for example, if it is cotton plant, this is crop. Okay, so in this crop we have introduced Cry1AC, Cry2AB gene, and what is target? What is target insect pest? Bollworm. So here you can focus on this line. Choice of gene depends upon the crop and the targeted pest. They are saying BT toxins are insect group specific. BT toxins are insect group specific. PYQ, insect group specific. Okay. So this was about BT cotton. Now students, in next paragraph, they are talking about pest resistant plant. Okay, BT cotton is also pest resistant plant. Now they have given one more pest resistant plant, and it is tobacco plant. Okay, one more pest resistant plant is tobacco plant. Okay, here they are talking about RNA interference. Sorry. In this paragraph, they are talking about RNA interference. So first, you can focus on this paragraph here. It is pest resistant plant. They are saying there are many nematodes which parasitize wide variety of the plants and animals. They have given one example, Melatogyne incognita. This nematode infect root of the tobacco plant. So this is one question. Nematode Melatogyne incognita infect which part of the tobacco plant? Root of the tobacco plant. Now this if this tobacco plant is infected, tobacco is a cash crop for farmers. If this tobacco plant is infected by nematode, it decrease yield of tobacco. Okay, so scientists, they have created new strategy. This new strategy, here they mentioned novel strategy. Novel strategy adopted to prevent this infestation was based on RNA interference. Now question is, how this Melatogyne incognita is created? What is novel strategy to control this nematode? Novel strategy to control this nematode is RNA interference. This is novel strategy. Novel means new. And student, this strategy basically it was discovered by Indian scientists. Okay, there was research on this plant. Okay, done at IIT Kanpur and one university is in Tamil Nadu. So they collaborated, they coordinated, and they developed this strategy. So they are saying this type of RNA interference is RNA interference. It is naturally present in all eukaryotic organism. RNA interference was discovered. We will study about this. It was discovered in one of the nematode. So they are saying RNA interference is present in all eukaryote as a method of cellular defense. As a method of cellular defense. Why it is called RNA interference? In this technique, we prevent expression of the mRNA. In RNA interference, this is important question. When we say RNA interference, so in this technique, we prevent translation of the mRNA. We prevent translation of the mRNA. Here they mentioned silencing of the specific mRNA. What is the meaning of silencing? It means mRNA will not form protein. It will not produce protein. Okay. It will not participate in translation. So how we will prevent its translation? Its translation, they are saying, is this is due to complementary double-stranded molecule. So we use complementary double-stranded RNA. We use complementary double-stranded RNA, and complementary double-stranded RNA help in silencing the specific mRNA. Okay. Now they are saying this type of strategy you will see. This type of strategy you will see during control of viruses naturally. During control of viruses in eukaryotes. Okay. I'll show you one diagram. So this type of strategy you will see during control of viruses, RNA viruses, and this type of strategy you will see preventing multiplication of the transposons that are replicated by RNA intermediate. Okay, I'll explain it how. First, you focus on this line RNA interference. Okay, in RNA interference, we prevent expression of the mRNA. We prevent translation of the mRNA by using double-stranded RNA. Okay, we prevent translation of the mRNA by preventing double-stranded mRNA. Now, this is one general diagram. You can focus on this. Suppose this is one cell. It is eukaryotic cell. Okay, this is eukaryotic cell. This is nucleus, and it is cytoplasm. Nucleus and cytoplasm. In cytoplasm in eukaryote, there is formation of RISC. RISC. What is present in RISC and what is RISC? RISC also called RNA induced silencing complex. Okay, RNA induced silencing complex. It means this silencing complex is activated by RNA. Okay, what are other components of this RISC? RISC have many types of proteins, and it is also associated with RNA. Okay, so this RISC is having many protein and it is having one RNA. Okay, if RNA is attached to this RISC, it is activated. It is present in cytoplasm. If this activated RISC is present here, and if there is a RNA complementary to this RNA, you can see this blue line. This blue line represents RNA. Okay, if this RNA or part of this RNA is complementary to other type of RNA which are present in cytoplasm. Now you can follow this black line. Okay, so you can imagine that this blue colored RNA is having complementary sequence. Okay, in black colored mRNA. So any mRNA which is found complementary to this blue colored RNA, it will try to bind or pair with this, and then protein which are present here, they act as catalyst, or they act as basically they cut this protein which are present here, they act as nuclease, they cut this black colored RNA. Again, in RISC there are two components, many types of proteins plus RNA. Okay, what is source of this RNA? I'll explain. So now imagine this is activated. It means there are many proteins in this, and these proteins are associated with this blue colored RNA. If it is like this, it is activated. In cytoplasm, if this activated RISC comes in contact with complementary RNA, it can bind with this complementary RNA and then cut this. Who will cut? Proteins. Proteins will be present in this. They are catalytic proteins. Okay, so they destroy this RNA. Okay. Now, question is, what is source of RNA for this RISC? This is a RISC. RISC is present in all eukaryotes. Okay, it is present in all eukaryotes. Okay, these are present in cytoplasm of all eukaryotes. But if it is not associated with this particular type of RNA, it is inactive. If it is associated with this particular type of RNA, then it is active. If it is active, then it can bind with complementary RNA and destroy this. Okay. If it is not active, then it will not destroy. Now, how it is activated? You can focus on this diagram. Okay, in NCERT they were talking about virus. They were talking about RNA viruses Okay, they are saying source of complementary or double-stranded RNA can be RNA virus. Okay, this line NCERT line. Now imagine this RNA virus, it infect eukaryotic cell. Okay, it infect eukaryotic cell. So now when a virus enter into eukaryotic cell, this virus multiply inside the eukaryotic cell. To multiply, it will replicate its genetic material. It will produce many RNA with the help of host. It use host cell machinery, and it produce many RNA and many protein particles, and then it increase number of the viruses. Okay, now imagine this virus is having double-stranded RNA. This double-stranded RNA, you know, virus. What is purpose of the? What is ultimate aim of the virus? Ultimate aim of the virus is to multiply itself inside the host cell. Okay, now this double-stranded RNA, it is converted by dicer. It is converted by dicer protein. By dicer protein, dicer is an endonuclease. This double-stranded RNA is digested by dicer protein. Now this dicer protein, it is called endonuclease. It cut viral DNA into small segments. Okay, it cut viral DNA into small segment. Now you can imagine this is having one blue colored part and black colored part. Now this blue colored part, okay, if it is coming in contact with RISC, it can activate RISC. Now this activated RISC in future, if it is coming in any, okay, viral mRNA, it is coming in contact with viral mRNA, then it destroy that viral mRNA. Okay, so no, you know, this virus entered host cell, it tried to multiply itself. So one strand of that viral mRNA or viral RNA is attached to RISC. RISC is activated, and if in future, if virus particles is trying to multiply itself, it produces numerous RNA. So that one strand of the RNA which is complementary to this blue RNA, if this strand come in contact with this RISC, then this RISC destroy viral RNA. Okay, so this is activated by viral RNA and it is destroying viral RNA. Okay, what kind of viral RNA? Double stranded. Okay, it should be double stranded. Now there is one more option. Okay, suppose transposons. In your book they are talking about transposons. They are saying transposons, they can also produce RNA intermediate, and that RNA intermediate can participate in RNA interference. How? First, what is transposon? These are DNA fragments. Okay, in transposons, how they multiply inside the cell. In your book, they mentioned they can produce RNA intermediate. So basically, in transposons, there are types. One type is retroposon. Retroposon is not given in your book, name not given. What is retroposon? Retroposon is a transposon. What is transposon? It is a DNA, mobile DNA. It's a DNA segment. Okay. So this DNA segment, how it multiply? To multiply, it can produce RNA. Okay. Then RNA can produce DNA. Now you can see this is transposon. It is present in nucleus. Transposon is present in nucleus. Now this transposon produce RNA. This is double-stranded RNA. From nucleus, it migrates into cytoplasm. In cytoplasm, with the help of enzyme dicer, it can be converted into fragment. Okay, so this is double-stranded RNA. You can see black strand and blue strand. Now with the help of endonuclease called dicer, it is converted into small segments. Small segment may be called siRNA. Now this small segment, from this small segment, one strand bind to RISC and okay, RISC. Now this RISC is activated. This RISC, now this blue colored RNA is complementary to one strand of this RNA produced by transposon. Okay, so in future, if this activated risk is coming in contact with this type of RNA which is coming, okay, which is synthesized by transposon, and it is complementary to this. Now this RNA which is produced by transposon is destroyed by activated risk. Okay. Now how it is related to this transgenic tobacco? In your book they are talking about transgenic tobacco. That transgenic tobacco, it is protected from nematode, and it is created by scientists. It is created by scientists through recombinant DNA technology. Now suppose this is tobacco. This is basically nematode. This is nematode. Inside a nematode, risk can be activated by artificially providing RNA to this nematode. Okay, this is inside nematode. Now you can focus on exogenous RNA. Exogenous RNA means it is coming from external source. Okay, what is external source for nematode? External source for nematode is plant cell. Suppose nematode is feeding on plant cell, and plant cell is producing RNA. How plant cell will produce RNA? We will discuss that. Okay, but plant cell is producing RNA. If nematode feed on plant cell, then RNA from plant cell enter into nematode. Now in nematode cytoplasm, this RNA may activate RISC. So this is double stranded RNA. In NCERT, they are talking about double stranded RNA. Again and again, they are talking about double stranded complementary double stranded RNA, which initiate RNA interference. So this is double-stranded RNA. It is produced inside plant cell. It is produced by plant cell. How plant cell will produce? We will discuss this. Now plant cell is producing double-stranded RNA. This double-stranded RNA may enter into nematode. Now one strand of this may bind with the RISC. Okay, and one strand of this may bind with the RISC. This RISC is activated. So this activated RISC may destroy nematode mRNA. Okay, now you can focus on this NCERT. Here they talk about pest resistant plant. It is pest resistant plant. It can control nematode. How to control nematode? By RNA interference. They are saying in RNA interference, complementary double-stranded RNA molecule, it bind to and prevent translation of the mRNA. Whose mRNA? In this case, this mRNA is of nematode. It is mRNA of the nematode. Its expression is prevented by this double-stranded RNA, and double-stranded RNA is synthesized by plant. Okay, and nematode is a parasite on plant. So when nematode feed on tobacco plant, inside tobacco there is double-stranded RNA that enter into nematode, and inside the body of the nematode, this double-stranded RNA, it activate a RISC, and that activated RISC, it prevent translation of the mRNA of the nematode. Okay. Now they are saying source of complementary RNA. It means this double-stranded RNA. Source of this complementary double-stranded RNA. It can be viruses having RNA genome, or it can be source of this double-stranded RNA can be mobile DNA element called transposons. Transposons are produced. Transposons, they are mobile DNA. They produce double-stranded RNA intermediate, and it can participate in RNA interference. Now these are two sources. They have mentioned here third source, but is source of double-stranded RNA nematode. Now they are saying scientists they have isolated nematode specific gene. Okay, scientists they have isolated nematode specific gene, and these genes are introduced in host plant, that is tobacco plant, with the help of vector. What is vector here? Agrobacterium. In agrobacterium there is a TI plasmid. That TI plasmid is modified by scientists, and it is used as vector. Now scientists they isolated nematode specific gene. They introduced this in TI plasmid of the agrobacterium. Now TI plasmid of the agrobacterium transfer these nematode specific gene into plant. Okay, there are two types of genes. These two types of genes, they produce two types of RNA. One sense RNA and second antisense RNA. Okay, so one gene produces RNA called sense RNA. This sense RNA is similar to nematode RNA, and second is antisense RNA. This antisense RNA is complementary to the sense RNA. So inside a plant, these two different types of gene, they produce two types of RNA, sense, antisense. Now inside plant cell, sense and antisense RNA, they fuse together and they form complementary double-stranded RNA. Okay, two types of genes are introduced in host plant, that is tobacco plant. Inside tobacco plant, these two types of genes produce sense RNA and antisense RNA. This sense RNA and nematode mRNA both are same, and this is complementary to double-stranded RNA. Both will fuse, they form double-stranded RNA. Now this double-stranded RNA, it initiate RNA interference. Okay, now again you can see this diagram. Inside plant cell, we have transferred two types of genes. Okay, two types of gene. One gene produce sense RNA, and second produce antisense RNA. Suppose this is sense RNA and it is antisense RNA. Okay, now they pair together and form double-stranded RNA. When nematode feed on host plant, this double-stranded from host tobacco enter into nematode. Now inside nematode, this one strand bind with RISC, it is activated. Second strand, which is called sense strand, it is similar to nematode mRNA. Now it is destroyed. Okay, now I told you this both of these genes are artificially introduced in tobacco plant. These are nematode specific gene. It means these types of genes, this type of gene is also present in nematode. So now when this risk is activated, suppose nematode is producing this type of RNA inside nucleus. Now this type of RNA from nucleus is transferred into cytoplasm in nematode. Now in nematode, this is supposed to synthesize protein, but for this RNA, risk is already activated. So when nematode synthesize this type of RNA in cytoplasm, risk is already activated. Now this risk will start destroying this sense RNA of the nematode. They are saying agrobacterium is used as vector, or modified TI plasmid of the agrobacterium is used as vector. With the help of this, we transfer nematode specific gene into plant. Plant produce sense and antisense RNA. It is similar to nematode RNA. Now they form double-stranded RNA. These are complementary, so they form double-stranded RNA. This initiate RNA interference. Okay, and this prevents translation of nematode mRNA. Students, this risk etc. is not given in your book, okay, and these are questions asked in NEET exam. You can focus here on question. They are saying this nematode Baladogyne, it infect root of the plant, and this RNA interference is present in all eukaryotic organism as a method of cellular defense, and this line was given in NEET exam, and it can be controlled by this in RNA interference we prevent translation of the mRNA with the help of double-stranded RNA. So you remember RNA interference is initiated by, this is a very popular question, RNA interference is initiated by double-stranded RNA. If they are asking how this translation of mRNA is prevented or silenced, by with the help of double-stranded RNA. Okay, double-stranded RNA. They are saying these transposons. There is one question, I think it was asked 2-3 years ago. Transposon, what is use of transposons? Transposons are used in gene silencing. These are used in gene silencing. Okay, you know transposons, they produce RNA, and that RNA can be used in RNA interference. So which vector was used to transfer nematode specific gene into plant? Agrobacterium or modified TI plasmid. Now these nematode specific gene inside host plant, they produce two types of RNA, sense, antisense. Now they are complementary, so they will form double-stranded RNA. Now this double-stranded RNA, it initiate RNA interference. So when we say that there is a RNA interference, it means mRNA expression is prevented or translation of mRNA is prevented. Okay, so students, this was related to BT tobacco. Okay, transgenic plant here is tobacco. In this chapter there are 4 topics. Topic 1, 2, 3, 4. 1 and 4 we will study in this video, and 2 and 3 you will study in next video. Now you can see this diagram here. Here they have given photographs of tobacco plant. This is transgenic tobacco and it is without transgene. It is not transgenic. So they are saying if both plants are infected by nematode, after few days you will see this develop root knot, and in this there is no knot. So it is not infected and it is infected. This non-transgenic plant is written as control plant. Control plant here is without transgene. Okay, it is infected by nematode, it develop root knot disease, and here it will not develop any disease. Okay, so this topic you will study in next video, and now you can focus on last topic 10.4. Topic 10.4, okay, ethical issues. So here they are talking about some ethical standards. Now suppose you have knowledge of recombinant DNA technology, you know procedure, okay, how to conduct experiment, you know this. So they are saying we cannot manipulate with genetic material of the plants or animals unnecessary. Okay, so there should be limit. We have to define boundaries to which extent we can manipulate with genetic material of the organism. Okay, so they are talking about ethical standards. These are required to evaluate the morality. Morality means these are rules which govern, okay, research, to evaluate morality of the human activities. Okay, we cannot unnecessarily we cannot change genetic material of living organism. Okay, so they are saying this modification of living organism or genetic material modification, it can have unpredictable result. Okay, so in India they have, government has set up one body, it is called GEAC. They are saying Indian government has set up organization, and this organization is called GEAC, and GEAC Genetic Engineering Approval Committee. What is use of this GEAC or this committee? This committee take decision regarding validity of the genetic modification, and it take decision regarding safety of introducing genetically modified organism. Okay, And this question was given multiple times in NEET exam. What is the role of GEAC? What is the role of GEAC? So in India, GEAC, they take decision regarding validity and safety of genetically modified crops. Validity and safety. And in one question here, they have not mentioned genetic engineering approval committee. They have mentioned genetic engineering appraisal committee. Okay, so sometimes they confuse students. Modification of living organism. They have created a problem with patent. How modification of genetic material created problem with patent? They have mentioned here. But first, you should know what are patents. What are patents? Patent is a legal right. It is a legal right granted for invention involving biological material, biological process, or biotechnological product, giving the inventor exclusive right to use, produce, and sell the invention for a certain period. Okay, suppose you are a scientist and you have created one genetically modified crop. So now you can sell this crop in market, okay, and government will give you legal right to sell this. So all economic benefit of that will be given to you for certain period, maybe 10 years or few years, okay. So how this biopatent has created problem? Now they have given one example here. They are saying in many countries like India, okay, these are biodiversity rich, okay, and there are many crops that we are using from many years, okay, or our ancestors have used those crops, and now private companies from some other countries, they remove gene of that crop and they introduce this in another crop, and they'll say that they have created this transgenic crop, okay. Basically they have given here one example. They are saying that one company, American company, in 1997 they got patent right on basmati rice, okay, through US Patent Trademark Office. How they created this crop? They created this variety by hybridizing Indian farmers basmati variety with American semi-dwarf rice variety. So what are two crops hybridized by them? Semi-dwarf varieties from USA and Indian basmati rice. They hybridized this and they created a new variety. Okay, and this private company, they claimed that they have invented this. They have invented this. But you know, basmati rice in India, we are using it from many generation. Okay, but these scientists, they created this crop and they said it is a new variety created by them, and then they applied for patent in USA and they got patent on this. What is meaning of patent? They got legal right to sell this new basmati variety in their market, okay. And now they are saying this patent extends to functional equivalent, implying other people selling basmati rice could be restricted by the patent. It means if we want to sell basmati rice in their market, we cannot sell this because their patent act will prevent this, okay. So they are saying this type of attempt has been made by many private companies from many countries, okay, like on turmeric and neem. Many companies, they tried patent on these product. So this is basically called biopiracy, okay. So they are saying they have introduced a one term, biopiracy. What is biopiracy? Biopiracy means it is use of bioresources by multinational companies and other organizations without proper authorization, without proper permission from the countries and the people concerned, without their compensatory payment. So you should know what is the meaning of biopiracy. So basically this is theft of bioresources, biopiracy. And then here they mentioned Indian Parliament has recently cleared the second amendment of the Indian Patent Bill. In India, you cannot apply for patent on traditional knowledge. Okay, if there is a traditional knowledge available in country, you cannot claim. Okay, you can apply. You cannot apply on patent. Okay, you cannot apply for patent on that traditional knowledge. Okay, so this is clearly written in Indian Patent Bill. They're asking question, can you suggest a method to remove oil or hydrocarbon from seed based on your understanding of the recombinant DNA technology? So for this, what is required? What are steps for this? First, you have to identify organism. First, you have to identify organism which contain gene and this gene is it synthesizes enzyme. That enzyme is capable of degrading hydrocarbon or lipid. So first you have to identify this gene in crop. Now suppose you have identified this this gene in one plant. You can isolate this gene and then you can join this gene to vector, and with the help of vector you can transfer this into desired crop. Okay. There is one more question. Does our blood have protease and nuclease? Answer is no. Now they're asking about how to make orally active protein pharmaceuticals. Okay. If you are using a protein as a pharmaceutical or drug and you are ingesting it through mouth, this is a protein, it is digested in our stomach and intestine. Okay, so if you want to use this protein as a drug, then you have to cover this in a gel. You have to pack this in a gel, okay, to prevent its digestion in alimentary canal, like in stomach. Okay, we discussed this topic number 10.1 and 10.2. Now we'll see some question based on this. Grain revolution succeeded in tripling food supply mainly due to, you can recall we studied here mainly and partly, so mainly it was due to better management practices. Okay, ability of plant cell or explant to generate whole plant is called totipotency. A plant produced through tissue culture or micropropagations are genetically identical to the parent plant. Such plants are called soma clones. These are sample question, okay, and you can read these question and options. Virus-free plants can be obtained from diseased plant by culturing the apical and axillary meristem. Somatic hybridization involves, in somatic hybridization, we fuse protoplast from two different plant varieties. How we prepare pomato? Pomato is a somatic hybrid. It is prepared by fusion of protoplast of tomato and potato. Which of the following is not advantage of genetic modification? You can recall that paragraph. There we studied through genetic modification, we can increase nutritional value, we can increase tolerance to abiotic stress, we can reduce dependence on chemical pesticide. Through genetic modification, complete elimination of all plant diseases is not achieved. All plant diseases not achieved. Okay. Bt toxin does not kill Bacillus thuringiensis itself. Why? Because in bacteria, this Bt toxin exists in inactive form, protoxin form. Gene CRY1AC and 2AB of Bacillus thuringiensis. These are transferred into cotton to control bollworm. Okay. Now, next question here is strategy used to protect tobacco plant from nematode. What is the name of the strategy? Novel strategy. It is RNA interference. In RNAi based pest resistant nematode specific genes were introduced into the host plant using which vector? Agrobacterium. Golden rice is a genetically modified crop enriched with vitamin A. What is full form of GEAC? Genetic Engineering Approval Committee. Biopiracy. What is meaning of biopiracy? Use of bioresources by multinational companies without proper authorization and compensatory payment. Biopiracy. Last question here. American company got patent right on basmati rice in 1997. This is an example of biopiracy. Okay. So students, from this chapter we completed topic 10.1 and topic 10.4. Thank you so much.